Hello everyone, it's your friend Ask Anything and how are you all? So in this video we are going to discuss about microscopy, different types of microscope and their uses. So now let's start our discussion with taking microscopy as the first point. So microscopy is the study of cells under microscope. Okay, so microscopy is the study of cells under microscope. So these cells are very small and they are generally in microns or nanometers. Okay, so now let's come to magnification. So what is magnification? So magnification is the ratio of the size of the image as observed under a microscope by the size of the image as seen by an unaided eye. So there is another formula which is for compound microscopes. It is power of objective lenses into power of eyepiece lens. Okay, so what are objective and eyepiece lenses? So I am drawing a rough figure of a compound microscope. It is something like this. So here is the point where we see. So there is a lens here which is known as eyepiece lenses. And when we go down here, the place where we keep our slides, just above that there are 3 or 4 lenses. So these are objective lenses of different parts. The maximum power of eyepiece lenses can be 20 times magnification, and the highest power of objective lenses can be from 200 magnification to 100 magnification. It can range between 1 to 200 magnification. So now let's come to the type of. So in microscopes there are 2 types of microscope. One is compound light microscope and another is electron microscope. So first we will discuss about compound microscope, and then we will proceed to electron microscope, which is further divided into transmitted electron microscope and scanning electron microscope. So in compound microscope, so compound microscope uses light usually sunlight to see the objects which is there in the slide. So and its magnification power is 300 times to 1500 times, and its resolution power. So what is resolution power? So suppose there are 2 objects and they are very close to each other. So it is the power of the microscope, or we can also talk about our eyes. It also has some resolution power. So the resolution power of our eyes is 100 microns. So what is my resolution power? So suppose there are 2 objects, as I told you, very close to each other. So it is the capability of eye on any microscope to distinguish them as 2 objects. For example, if we will get very closer, so eyes loses the capability to see those 2 objects as different. So this is known as resolution power. So the resolution power of compound microscope is 0.25 micron to 0.3 microns. Okay, so it uses light. So how it uses light? So there are 2 parts in the compound microscope which helps it for focusing light. One is condenser and the other is a reflecting mirror. Okay, so now let's proceed to electron microscopes. So electron microscopes are further divided into 2 types, TEM and SEM. So before proceeding, let us see who invented the electron microscope. So it was invented by Noel and Ruska. Place is Germany. The time was 1932. Okay, so it was invented in 1932 by Noel and Ruska in Germany. So now let's talk about these 2 microscopes. The full form of TEM is transmitting electron microscope, and the SEM full form is scanning electron microscope. So electron microscope uses a beam of electrons which has 10,000 times less wavelength than the visible light. See कितना difference है visible light और इस इलेक्ट्रॉन माइक्रोस्कोप के इलेक्ट्रॉन्स के बीच में 10,000 times कम है. So first proceed to TEM. So it catches or it uses transmitted electrons. Transmitted electrons, and SEM uses scattered electrons. Now, the function of these two microscopes, TEM and SEM, are very different from each others. So TEM delivers a two-dimensional figure, a 2D figure or 2D image of the object, because it is used to see what is beyond the boundaries of the cell, whereas SEM delivers a 3D image, but it is used to see the surface, the boundaries of a cell, but TEM is used to see beyond the boundaries of the cell. So the magnification power of TEM should be much high because it is used to see beyond the boundaries of the cell. So it is 50 million times. 50 million times. And the resolution power is 0.58. So now let's talk about the magnification and resolution power of a scattered electron microscope. The magnification power of SEM is 2 million times. See the difference. And the resolution power is 0.4 nanometers. The resolution power is also much less. So this was all about the microscopes, types of microscope, who invented the microscopes, and uses. So this was all about microscopes. So thank you friends for watching my video. Please like, subscribe and share my channel.